second part of the chapter hepatitis vaccine and edible vaccine, and in this part we'll learn about edible vaccine. Okay, so I mean, in the previous part, we saw that vaccine can be produced by using cell, right? Like microbial cell or mammalian cell. Well, theoretically, actually, in practical, scientists are also trying, and already they have been successful in producing antigen, that is vaccine, using plant. Okay, so what is the strategy in this case? So, like the basic state, the first few steps will be same. First, the gene, first, suppose you want to uh, you want to produce your vaccine candidate, that is the antigen in a plant. So first you need to select that which antigen you will use as a vaccine. Then you have to identify the gene. Okay, so once you identify the gene, then you can you can just get the gene, right? You can use there are molecular methods. You can get the gene from the pathogen and can amplify by PCR. Okay, then you have to insert this gene into the plant cell. Okay, so this can be done by a process called transformation. So once you insert the gene into the plant cell, that gene will get integrated into the DNA of the plant cell. Okay, and the new plant, will be called transgenic plant. That is, the transgenic plant is that plant which has incorporated gene into its genome. Okay, so then once you get the transgenic plant, so whenever this plant will grow, they will also express the express your express the gene that will produce the antigen. Okay, so and that antigen will be a part or component of that plant. So whenever the host, for example, it's a human vaccine, so whenever the human will eat that plant, they will get the antigen. So once, so as they will get the antigen, then this antigen can act as a vaccine, because this antigen can then induce immunity in the host body. Okay, so as this kind of vaccine can be eaten, so that's why these are called edible vaccine. Okay, so actually, scientist has, so far scientist was successful in producing different antigens. The antigens of different pathogens using plant. Okay, so just for example, the first human clinical trial took place in 1997. It was a vaccine for E. coli. Okay, actually not the E. coli. It was a vaccine against the toxin produced by E. coli. Okay, and it was uh, produced in potato. And ingestion of this transgenic potato resulted in satisfactory vaccinations and no adverse effects. However, actually, in this case, only the phase 1 trial was done. Okay, so yes, it didn't go for phase 3 trial. Okay, so because still now, the edible vaccine things sounds very nice. I will say that, because we'll produce the vaccine in the plant and we'll just eat them and we'll get vaccinated. Okay, it sounds really good, but the fact is there are many unanswered questions here. Because unanswered questions means, for example, one question is whether that antigen will be stable in that plant. Okay, or what will be the amount of antigen in that plant. Still it's not actually possible that the amount of antigen will be eventually evenly distributed in all the parts of the plant or in all the parts that we eat. Okay, and also there are unanswered questions regarding the safety issue. Okay, so still now all the edible vaccine, they are under their experimental stages. Okay, and not only this one, several other edible vaccine was also produced, such as in 1984 transformation of tobacco. uh, in the research for making edible vaccine, the first transgenic plant was tobacco. Okay, okay, and in the process of developing edible vaccine, once you identified your gene. So now this gene need to be transferred into the plant. Okay, so this can be done by, basically there are two ways to do this. One is agrobacterium mediated transformation and another is particle gun bombardment, which is also called in short ballistics. Okay, so first, agrobacterium mediated transformation. Okay, so what is done here? So in this photo, basically actually it shows both the agrobacterium mediated transformation process and also the gene gun or ballistic methods. Okay, so I will talk about the ballistic methods later. First, just concentrate on the left side of this photo. Okay, so agrobacterium mediated transformation. So what? First, you need to identify the gene that encode your antigen. Then you have to isolate that gene. Then insert that gene into a plasmid. Okay, so in case of agrobacterium, the TI plasmid is a good option. Okay, because this TI plasmid, it expresses quite well in the agrobacterium. Okay, so you got your gene, you identified your gene and isolated it and inserted into the plasmid. That is, you now have your cloned plasmid. Then you have to insert this plasmid into the then actually you have to insert this, I mean you have to uh, okay, so actually you have to insert this plasmid into the bacteria, agrobacteria, and then insert transform that bacteria into the plant cell. Okay, so that means actually in this case the agrobacteria, it will help you to transform the plasmid into the plant cell. Okay, so once the plasmid is inside the plant cell, the gene, I mean your gene of interest, it will get integrated into the genome of the plant cell. Okay, and in this process, I mean all the plant cell will not I mean gene, your gene of interest will not incorporate into all the plant cell. Okay, so actually, so after this step, okay, so you have to screen, so you have to find your transform cell. Okay, because all the cell, I mean the your gene of interest will not get integrated into all the cell. So through screening process, you have to find out the cell, the transform cell, that is which has your gene of interest. So from that cell, when the transgenic plant will grow, it will I mean, as this transgenic plant has integrated gene in its genome, so it will also it will express that gene and it will produce the antigen. Okay, so this was the steps, basic steps involved in producing transgenic plant. I mean, edible vaccine producing transgenic plant. Okay. Okay, so now particle gun bombardment method. Okay, so another method of transformation of gene into the plant cell. Okay, so in this case, okay, so particle bombardment or ballistic and microprojectile bombardment is a direct method of gene transfer. I mean, in case of agrobacterium mediated transformation, the gene is first inserted into the, I mean first cloned into the plasmid, then this plasmid is inserted into the agrobacterium, then this agrobacterium is inserted into the plant cell. Or say it's kind of a indirect method, but in case of ballistic method, gene is directly transferred into the plant cell. Okay, so how it is done? So this process involves the use of heavy metal and inert particles such as gold or tungsten coated with DNA to be transferred into the plant. This DNA is your gene. Okay, and heavy metal and inert particles that coated with genetic materials are known as microcarriers. Okay, and these microcarriers will be transferred with high speed into the plant cell. Okay, so this photo shows the ballistic procedures, such as this is the gene gun. So you have to use particles, a small particles of gold and tungsten. Okay, and you have to coat these small particles with your gene of interest. Okay, then you have to put this gene of interest in plastic disc, and through the gene gun, uh, this plastic disc will be, I mean, you have to shoot this plastic disc into the plant cell. Okay, especially actually like if you see this photo, this is the gene gun. Okay, those, I mean, through this gene gun, this plastic disc which contain the DNA coated gold particles are shoot to the leaf of the plant. Okay, so at in high speed. Okay, so in that procedure, those DNA coated particles, because of the high speed, they will insert into the plant cell. Okay, so once this I mean, DNA coated particles are inside the plant cell, some of the gene will get incorporated into some of the plant cell. Okay, and then again through a screening procedure, you have to find out the cell which is, I mean, you have to find out the transform cell, and from that transform cell you have to get your plant. Okay, you have to develop your plant. Okay, okay, so well, so I mean, scientists are also trying to, I mean, some scientists are also trying to develop hepatitis B vaccine using this strategy. That means they are trying to produce edible hepatitis B vaccine. Okay, so the basic methods is same. So the basic methods is same. I mean, like if you use agrobacterium mediated method, you will use this method. If you use gene bombardment, you will use this method. Okay, just what will be the only difference? The difference will be your gene. So if you want to make a edible vaccine for hepatitis B, so you have to select your gene for hepatitis B antigen. If you want to develop a vaccine for cholera, you have to select the gene of cholera antigen. Okay, well, in case of hepatitis B surface antigen, some scientists, they actually also tried an additional and a different approach. Different approach means they tried to use virus, okay, to generate the edible vaccine. So what was the role of virus here? I mean, in case of agrobacterium based transformation, basically what was the role of agrobacterium? The agrobacterium is used to transfer the gene into the plant cell. So if we use virus, so the role of virus here is to transfer the gene into the plant. Well, okay. So actually, what is the difference? I mean, why some people used to try? I mean, some people, some scientists, they try virus to transfer the gene, because some virus, I mean, they can infect the plant cell more easily. Okay. So and we know once a virus it infect a cell, it's actually inject its DNA into the cell, right? Okay, so theoretically, if you can uh, produce a mosaic virus which contain your gene of interest, and then you use this virus to infect the cell, so after infection, that virus will inject its chimeric DNA into the plant cell. Okay, so that means the whether you are using the agrobacterium or you are using the virus, basically you are using the bacterial virus just to transfer the DNA into the cell. Okay, okay, so this table it shows some example antigen of different pathogens for which uh, transgenic plant is developed, already developed. Okay, so however, although scientists have already generated different transgenic, I mean, edible vaccine producing transgenic plant, but still now no edible vaccine is in the market. So hopefully in future, okay, we'll get those kind of, I mean, we'll get vegetables or fruits, okay, which will have the antigen in them. Okay, so it will serve two purpose, right? We'll get the nutrition from the food and vegetable, and also at the same time, just consuming those food and vegetable will get vaccinated. Okay. Okay, so that was all about the hepatitis vaccine and edible vaccine. But before finishing, I want to give you an idea about the cold chain. Okay, so you will actually frequently, if you work in the I mean vaccination field, so you will frequently heard this word cold chain. So what is cold chain? Well, we know that vaccines, okay, so most of the vaccines are biological particles, right? So either they're proteins, or sometimes they're conjugate vaccines, right? The carbohydrates mixed with proteins, okay. So these are biological molecules, okay, so they need to be properly stored, okay. So actually, most of the vaccine, you just cannot keep them in the room temperature, because if you keep them in the room temperature, there is a chance that they will get degraded, okay. So that's why you have to always, I mean, in storage, in transportation, okay, always you have to use cold temperature. So this is called a cold chain, okay. So for example, in the factory, they are producing the vaccine, so after producing, they have to store it in cold temperature. Then when they will transfer it, they again have to transfer it, maintaining the cold temperature, okay. So this is called cold chain, maintaining cold temperature from the beginning to the end, okay. So, and the, I mean, different equipments are used for cold chain maintenance. For example, I mean, we can just use refrigerator, okay, but the refrigerator, they have certain limited capacity. So somehow, instead of just a refrigerator that we see in our home or in the different shops, cold rooms are used, okay. So if you can, actually, in many different institutions and storage facility, they have large cold rooms, okay. So if you, in the cold rooms, I mean, their capacity is much higher than the usual refrigerator, okay. So cold rooms are used, uh, freezers, okay, both refrigerators or freezers are used, okay. And for transportation, if you need to transfer a small amount of vaccine, you can use cold box. If you need to transfer large volume of vaccine, you can, like, you can use refrigerated car, okay, or freezer car, okay. And I mean, if we think about the cold chain, okay, so we cannot actually store, I mean, at the same temperature will not be applicable for all the vaccines, okay. So different vaccines, they have different requirement of cold temperature, such as, for example, polio vaccine, okay. Polio vaccine is a very heat sensitive, okay. So we need to store them at -20 degrees Celsius, okay. But some vaccines, for example, typhoid or DPT, we cannot freeze them, we cannot keep them as -20 degrees Celsius. So basically, we have to store them like around 4 degrees Celsius. I mean, we have to store them at cold temperature, but we should not never allow to freeze them. So if they freeze, I mean, like for example, typhoid or DPT vaccine, if these vaccines get freezed, those vaccine will be damaged, okay. Okay, so that was all. Thank you for your patience.